Evening. Please. So, now I'm going to talk about immunologic tolerance and then a little about autoimmune diseases, or the basics of autoimmunity, not the autoimmune disease indeed. What does immunologic tolerance mean? What does immunologic tolerance mean?
Our immune system don't attack our own antigens on
Yes.
body.
Yes, tolerating self-antigens, self-tissues, which they are called autoantigens. So, tolerating self-antigens ensures that in normal situations, we won't have autoimmunity. Without tolerance, we will have autoimmunity. So, I'm going to talk about how we have tolerance induction in T cells and B cells. In the last lecture, I told you that billions of TCRs and BCRs are produced in B cells and T cells in the bone marrow and thymus. And I told you that there is no antigen exposure, foreign antigen exposure in the thymus and bone marrow. Please, it's very important question for that T cells and B cells without exposure to foreign antigens, they make billions or millions of TCRs and BCRs. For example, when you haven't exposed to Zika virus, but your immune system has TCR or BCR against Zika antigens. So you haven't exposed to Zika virus yet, or Ebola yet, or HIV yet, alhamdulillah. But your immune system has TCR and BCR, or specific clones against Zika, Ebola, and HIV.
By vaccine?
No. Have you received HIV vaccination? No. Have you received Ebola vaccination? No. But you have, I didn't say that you have active immune response. You have TCRs and BCRs. You have clones, naive clones against them, that they haven't been exposed to their own antigens. When we receive vaccine, indeed we are exposing the clones to the antigen, and then we won't have naive T cells or B cells. We will have memory. The aim of vaccine is to induce memories, T cells and B cells against different antigens. But the story that I'm telling you now is different. How is it possible? How is it possible that you haven't been exposed to Zika virus, but you have TCR and BCR against it? It's antigens.
Because of the mechanism that we discussed in the previous session about combination and diversity and junction of diversity. So by this mechanism we can have a lot of receptors in different shape that can and also about the tolerance and T cell and B cell during the development see a lot of self-antigen, and if the interaction was in high affinity, will get to apoptosis and death. So by that we can prevent autophagy.
Yes, thank you. What else?
By continuous change in the...
No. It's very important. There is no change in the TCRs and BCRs. There is no change. You have millions of clones, they are ready to respond, always.
Because I think there are changes...
Please?
I think there are...
Actually, yeah?
There are continuous changes in the sequence of the amino acids and the cells. maybe from the cell to the other cell. From the mother cell to the other.
No.
It's the
I
same
told you. We haven't received this these huge clones from our parents. We just have received that genes, but the recombination has occurred in your body. No, don't. What? You want to say something? He says that we might inherited these clones from our parents. No, it's not. The only thing we have inherited from our parents are the genes, the fragments. And then in our body, we have recombination to produce billions or millions of TCRs and BCRs. It's simple. Immune system for production of TCR and BCR doesn't need to expose to the antigen. It will produce randomly millions of TCR. Millions of TCR randomly. That's why we need millions or billions of TCR. That some of them is possible to recognize one antigen. There is a good example of lock and key. For example, there is a lock here that I do not have any key for that. And I'm going to open the lock. I will make thousands of keys without seeing that lock. But one of the keys, it is possible that might be able to open the lock. If I have millions of keys, the possibility will increase. If I have billions of keys, the possibility will increase again. Is that clear? So, without exposure to foreign antigens, I told you, in the primary lymphoid organs, in the primary lymphoid organs, there is no foreign antigen. All the antigens are self-antigen. There is no foreign antigen. It's not possible to be exposed to all antigens of the nature, all viruses, all microbes, all tumor antigens. It's not possible. It's absolutely impossible. So immune system during the evolution, it has evolved in a way that can produce randomly billions of TCR. That some of this PCR is possible to recognize any antigen. Okay? Clear or not? So, for example, when you go to a clothing shop, in the clothes shop there are many types of clothing. Without seeing you, they have produced a specific type of clothing that it is fit to you. Okay? It is made without seeing the antigen, and it is made randomly. But the numbers, the huge numbers, will cover any possibilities in the nature, from microorganisms, tumor antigens, and any antigens in the nature. That's why it is important when T cell develops or B cell develops in the bone marrow or thymus, they produce billions of T cells. And I told you that, for example, in the cortex of thymus, there are so many huge, dense population of the T cells. But when we go from the cortex to the medulla, the population of T cells are not dense over there, because 97% of the T cells has been eliminated by apoptosis. Because TCRs are being produced randomly, some of them are useless TCR, and some of them are autoreactive TCR, and they have to be removed. So when we have random production of TCRs, so it's possible that some of them might recognize self-antigens, okay? And some of these TCRs might be useless at all. So those TCRs or those T cells having useless TCR or autoreactive TCR have to eliminate it from the thymus. They are not allowed to mature and leave the thymus or bone marrow for B cell, because the process is random. Okay, is that clear? So it's, you will understand how immune system would be unresponsive to self-antigens. factors can influence self-tolerance, induction, and maintenance, and how does immune system maintain unresponsiveness to commensal microbes. I'll talk about it tomorrow in barriers immunology. And so, it is important to know that immune system is not allowed to induce immune response against self-antigens. It's simple, because there are self-antigens. Not only immune system shouldn't induce any response against self-antigen, it shouldn't induce immune response against any harmless antigen. For example, normal flora. We need normal flora in our gut. Okay? So, immune system shouldn't eliminate normal flora from the gut or GI tract. Microbiota. On the other hand, we are exposed to some antigens in the air that they are not harmful for us. For example, pollen or dust. We are inhaling dust, but dust is not pathogenic. It's harmless. So we shouldn't induce immune response. We do not need to have immune response against dust, for example. On the other hand, we are exposed to food antigens. They are foreign antigens. If you inject food directly to the intravenously, it's possible to induce shock in you as a result of severe immune response. But we are exposed to food antigens in GI tract without immune response. So immune system has to only respond to not all microorganisms, just pathogenic microorganisms. For example, HPV, HIV, E. coli, and many others. Not all microorganisms, because most of them are not pathogenic, are normal flora or microbiota of the GI tract. And immune system, there is no need to react to self-antigens. The only thing that we need to react is microorganism, pathogenic microorganism, and tumor cells. That's it. That's it. The rest of the antigen, when the antigen is harmless, there is no need to have immune response against it. So we need to have tolerance against self-antigens, normal flora antigens, food antigens, with different mechanisms. But this lecture is about self and the antigens of the fetus by mother. The antigens of the fetus. The fetus has many antigens that are foreign to the mother, and mother shouldn't respond to the antigens of the fetus. If mother induces immune response against the fetus, the mother will have abortion. Most of the abortions are made by immune system in the mothers that cannot control the tolerance against the fetus. So we need to have tolerance against fetus antigens, food antigens, harmless airborne antigens, normal flora. The only we have to induce immune response in two conditions: pathogenic microorganisms and tumor. Okay, so in order to have tolerance against the antigens, mainly self-antigens, the topic of this lecture, the immune system tries to exclude, eliminate self-reactive receptors as much as possible, but it cannot eliminate all self-reactive TCRs and BCRs. I'll talk about that. And immune system has access to self-antigens continuously. When we have tolerance to self-antigens, it doesn't mean that immune system doesn't have access to the antigens. Of course, it has access to the antigens. There is another mechanism that while the immune system has access to the antigen, but some mechanisms prevents the activation of the immune system. And first of all, prevents the production, development of self-reactive T cells and B cells, and then some other mechanisms prevent the activation of self-reactive T cells and B cells. Okay, so my commensal microbes, I'll talk about this in tomorrow's lecture. And I told you that immune system in pregnant female is very important to tolerate the fetus. And again, we need to compare toleration versus immunogen. What is the difference between them? Toleration, immunogen.
Immunogen, antigen or substance that generate immune response. Tolerogen, unresponsiveness.
Yes, as the name shows, tolerance generation. Immunogen, immune response generation. So immunogens are the antigens that can induce immune response. For example, antigens of pathogenic microorganisms. But the tolerogens are the antigens that immune system is unresponsive to them, or they can induce unresponsiveness. They cannot induce immune response. It depends on the nature of antigen. I will talk about it. So, as immune response is specific, the tolerance is specific as well. The tolerance, it's important to keep in mind that when we talk about tolerance, we are talking about T cells and B cells, adaptive immune cells, not macrophages, dendritic cells, and the rest of the cells. They are indirectly involved, but not directly. When we talk about tolerance, we are mainly dealing with T cells and then B cells. Okay? Tolerance occurs in T cells and B cells. That's why I'm talking about TCRs and BCRs, not PRRs. In development of PRRs is different. We have both T cells and B cells are involved. So, knowing the mechanisms of immunologic tolerance may help us to develop some therapeutics for the treatment of autoimmunity, for tissue transplantation, for prevention of abortion, in cancer treatment. You will see that sometimes cancer uses tolerance mechanisms in order to evade from immune response, to escape from immune response. So if we understand the mechanisms of tolerance, we can develop some ways in order to activate immune system against a tumor that now are being used in the clinic against different tumors, different cancers. We have different approaches of immune therapy that is being used to treat cancers by knowing these mechanisms. So they are not just basics that are useless in the clinic. We use them in the development of therapeutics, both in reproductive, in cancer treatment, in autoimmunity, in allergy, in all of them. There are two main types of tolerance: central tolerance and peripheral tolerance. Central tolerance is induced in developing lymphocytes, where the development of lymphocytes occur in the body. Where does the so it occurs in the primary or generative or central lymphoid organs. There are different terms for same thing: generative lymphoid organs, primary lymphoid organs, or central lymphoid organs. That's why it is called central tolerance. So central tolerance occurs in developing T cells and B cells. Indeed, in central tolerance that occurs in developing T cells, some mechanisms try to prevent production of self-reactive T cells and B cells as much as possible. Okay? some mechanisms prevent the production of self-reactive T cells and B cells. But it's not successful 100%. Always we have some self-reactive T cells and B cells that can leave the thymus and bone marrow. That's why we need another form or another state of induction of tolerance, which is called peripheral tolerance. Peripheral tolerance occurs in the peripheral tissues, peripheral lymphoid organs. And so here it occurs in developing lymphocytes, but here it occurs in mature lymphocytes. Not developing, not immature lymphocytes. When we say developing, it means that they are not mature. They are immature lymphocytes. And it occurs in secondary lymphoid organs and peripheral tissues. So, indeed, the peripheral tolerance completes the action of central tolerance. We need both of them. Peripheral central tolerance prevents the production, but it's not successful. Always, we have some self-reactive T cells that are circulating throughout the body, so we need peripheral tolerance to prevent their activation. So, in the central tolerance, prevention is on the production of TCRs and BCRs that are self-reactive, but in peripheral tolerance, it prevents the activation of the clones. Is that clear? In central tolerance, the main aim of central tolerance is to prevent the production of self-reactive T cell and B cell. It prevents the production of T cells and B cells that are reactive against self-antigens. But in peripheral tolerance, there are mechanisms that prevent the activation of the clones. It prevents the production of self-reactive clones, production or development. Here it prevents the activation of the clones. Because we have the clones inside the body. Is that clear or not?
It prevents the central.
It cannot prevent completely. Some self-reactive clones can leave the thymus and bone marrow. It cannot fully eliminate all self-reactive antigens. I'll tell you why. There is a logical reference for that, reason for that. No question? Do you know the difference between central tolerance and peripheral tolerance in terms of the cells that are undergoing tolerance, the location of the tolerance, and the main aim? All of them are important. So, in central tolerance that occurs in bone marrow and thymus, the main focus is on the production of the T cells, to prevent the production of. But in the peripheral tolerance, some clones, some self-reactive clones that has evaded from the mechanisms of central tolerance, they are controlled in the peripheral tissue, or the activation is inhibited by some mechanisms in peripheral tissue with different mechanisms of central tolerance. So, you know the difference between naive and effector lymphocytes? Yeah? Both in terms of their properties, definition of them, and their activation. What is the difference between activation, requirement for naive and effector T cells or B cells?
In the cells which inactive.
Properties?
Yes.
No, inactive, no. Yes, they are. It doesn't mean that they are inactive. No. Definition.
Effector T cell doesn't require co-stimulation.
For activation, effectors, naive T cells need co-stimulation, but effector T cells do not need co-stimulation. Definition? What's the definition of naive?
B cell or T cell, it doesn't recognition the antigen.
Yes, any T cells and B cells that hasn't recognized its own specific antigen. That's all. A mature T cell and B cell that has left the bone marrow or thymus and it hasn't been exposed to its own specific antigen. It's the definition of naive T cell, but after recognition of the antigen, it's a specific antigen, it will differentiate to effector T cell or B cell.
Also, naive T cell, have memory, but effective have memory.
No. In naive T cells,
Both of them don't have
naive T cells or B cells, after activation, most of them differentiate to effector cells, and some of them differentiate to memory cells. They are different cells. It doesn't mean that effector cells have memory. They are different cells. Okay? So, knowing this is important for peripheral tolerance induction. Now, central tolerance in T cells occurs in the thymus. We have different phenomena inside the thymus that prevents the development of try to prevent development of self-reactive T cells. So it occurs in immature and developing lymphocytes. Again, inside the thymus, we have a stem cell. In different stages, it is undergo development toward a mature lymphocyte. And different scenario is possible for T cell in the thymus, for developing T cell in the thymus. So, it's important to know that in the thymus, we have developing T cells that they are producing T cells, TCRs randomly. They can produce any TCRs they want, for example. Okay? And besides, they are exposing to self-antigens in the thymus. They are exposing. There are some APCs in the thymus. Some types of endothelial cells and some dendritic cells in the thymus, they present antigen, self-antigen to developing T cells. Okay? To check if this developing T cell is self-reactive or not. So we need to have some self-antigens in the thymus to check their reactivity against them. So, for example, if I am a developing T cell, okay, if I am a developing T cell in the thymus, I'm exposed to several self-antigens, not one. If I cannot recognize, for example, they are all self-antigens. If I cannot recognize them one by one until maturation, I'm deserved to leave the thymus because I'm not self-reactive. But if I recognize just one of them, for example, as this is my TCR, recognize this strongly and become activated, then it means that this T cell is a self-reactive T cell. It has to be eliminated. Okay? The main process is this, but several scenarios happen in the thymus. So, first of all, we are developing T cells. They are producing TCRs randomly, many types of TCR. When we say many types, we mean TCRs with different specificities. They are producing them, and they are exposed to self-antigens with the presentation of antigen by APCs in the thymus. So we have APCs in the thymus. Let me ask a question. If a person doesn't have APC inside his thymus or her thymus, what will happen to him or her? Autoimmune. When there is no APC in the thymus, there is no antigen presentation. So there is no checking. Okay? There is no checking for T cells. When there is no checking, it's possible that so many self-reactive T cells can leave the thymus. So without MHC, the possibility of, without APC, without APC, the possibility of autoimmunity will increase. So, now, when T cells have TCR, this is TCR, and for example, this one is, I think, CD4, and this one is CD8. Developing T cells, when they are immature, they have both CD4 and CD8. So they are called double positive T cells. Double positive T cells means an immature T cell that has both CD4 and CD8. It's an immature T cell, it's not a mature T cell. We have them only inside the thymus. Okay? So this double positive T cell is exposed to self-antigen. In the first scenario, or let me explain it from this one. If in the first scenario, if the T cell can recognize the antigen strongly, or if binding of the TCR to the antigen produce a strong signal, it means a strong recognition. So it means that this T cell is self-reactive. It's against this peptide, and this is a self-peptide. This is self-peptide, this is MHC, this is TCR, and this one is, for example, dendritic cells or thymic endothelial cells. So, T cells is binding to self-peptide and self-MHC. If this T cell can recognize and induce a strong signal or a strong recognition, it means that T cell is self-reactive. So it has to be eliminated by apoptosis. It is called negative selection. That's simple. We have a T cell that it binds to self-antigens firmly and produce a strong signal throughout the cell. So this is a self-reactive T cell and has to be eliminated. And it is eliminated by apoptosis, which is called negative selection. Okay? Clear or not? Another mechanism is this one. We have a TCR. That this TCR cannot make any recognition. When we say it cannot make any recognition, it means that this TCR cannot be fitted to self-MHC. It cannot recognize self-MHC. You know that TCR needs to bind self-MHC in order to recognize the peptide. If a TCR cannot bind to self-MHC, it cannot recognize the peptide that is on the cleft of MHC. So when a TCR cannot recognize self-MHC, what does that mean? It means that this T cell is a useless T cell. It cannot produce any immune response. Yes, it's a useful T cell. If this T cell leaves the thymus, in the periphery, it cannot recognize any antigen. Why? Because it cannot bind to self-MHC. The first requirement for T cell activation is binding of TCR to MHC, and then recognition of the peptide in the cleft of MHC. If a TCR cannot bind to, for example, if this is MHC and this is self-antigen, or any other antigens, any types of antigen. If this TCR cannot bind, for example, to this MHC, it cannot recognize this peptide as well. So the TCR has to be able to bind to recognize self-MHC in order to be able to recognize the peptide. So this TCR is a useless TCR, and that TCR is a useless T cell, and it has to be eliminated again and again. We have apoptosis. It is called failure of positive selection. Failure of positive selection. Is that clear? Two scenarios. The first one, self-reactive T cell. The second one, a TCR that cannot recognize any MHC, self-MHC or self-HLA. There is another scenario that TCR can be matched with MHC, can be fit to MHC, but it cannot recognize the peptide. So there is just a weak recognition. This recognition comes from binding to MHC, not the peptide. This strong recognition comes from binding of the peptide and MHC together. This no recognition means there is no fitness to the MHC by TCR. So when there is a weak recognition, it means this TCR is fit, is matched to self-antigen, and it will induce a weak signal. And this weak signaling, weak recognition is enough to allow the T cell to become a mature T cell and leave the thymus. Okay? This is the third scenario. And this scenario happens for both CD8 positive T cells and CD4 positive T cells. For both of them. No question? This is called positive selection. So positive selection means selecting of the colons that are useful colons. They can match with MHC, but they are not self-reactive. They are not self-reactive against what? Against the antigens that are expressed in the thymus. That are expressed in the thymus. They are not self-reactive against the antigens that are not expressed in the thymus. So, is that clear or not? They are the process or scenarios of central tolerance in T cell. But there is a question. When we have central tolerance, why the centrals cannot eliminate all self-reactive T cells? The question is this: why does central tolerance, or why doesn't central tolerance eliminate all self-reactive clones? It can eliminate some of them, not all of them. That's why we need peripheral tolerance. It's very important topic, please. All self-reactive T cells is not possible to be eliminated in the central tolerance. Just some of them are eliminated. That's why we need peripheral tolerance to complete the action of central tolerance.
Because you don't have the thymus, you don't have all antigens of the body.
That's okay. Yes, we do not have all antigens of the body inside the thymus. We have just some of them. So we have checking against some self-antigens, not all self-antigens. T cells that are autoreactive against some self-antigens, that's why I told they are non-self-reactive against the antigens that are expressed in that are produced, that they are in the thymus. Okay? Clear? It's not possible to have all antigens in the thymus. For example, the antigens of skin, there is no antigen of skin in the thymus. Or antigen of the liver, or antigen of the spleen cells, or antigen of the neural cells. It's not possible to have them in the thymus. In the thymus, we have general antigens, housekeeping, and what does housekeeping genes mean? Housekeeping genes are the genes that their product is needed for basic life cycle of all cells. For example, genes that their product are needed for transcription, translation, ATP production, metabolism, all of these are housekeeping genes. They are needed for basic life cycle of the T cell, all cells. We have housekeeping genes are expressed in all cells of the body because they are needed for the basic biology of all cells. Okay, so mainly based the housekeeping genes and some other genes. We have thousands of genes. Around we have, for example, 100 different proteins, and each protein have different epitopes. It's not possible to have all of them in the thymus. Yeah? Clear? It's very important question.
You need to have to label this toxic unit. Can I label this? Supply this side?
Yes.
Yes.
You need to explain it again?
Yes.
Okay. All the process.
No, me explain this to.
Yes, yes.
Yes.
You want to explain?
Yes.
Yes.
The unit. you want.
You want me to explain or you want?
If you want.
He asked you if you want to explain. If you want.
If you want to.
You want to explain or you want me to explain?
No. I want.
Explain please. In Arabic or in Persian?
In Arabic.
Ah, yeah, please. That's good. Two group of cells.
Okay, so what's the self-antigen that shout the T-cell through the development? Self-antigen in the
Thymus.
yes, that show the T-cell during the development. You say that not all self-antigen.
Some of them are expressed in the thymus, not all of them. So what's your question?
Some of them, what's the
I do not know. We do not know which antigens are expressed in the thymus exactly. You want the name of the antigens? Several or many antigens are expressed, but not all of them. And we do not know that. Be sure that housekeeping proteins are expressed. All housekeeping proteins. Housekeeping proteins are the product of the genes that their product is needed for the basic life of the cell cycle of the cell. Okay, and some others. There is a gene in the thymic cells that enables the APCs to present, for example, antigens of the thyroid over there, antigens of the kidney over there, but not all antigens. It's not possible to have all antigens in the thymus. So when we do not have all antigens in the thymus, we do not have checking against all of them in thymus. Just some autoreactive cells are eliminated. Not only autoreactive cells are eliminated, useless T cells are eliminated as well. Two types of cells are eliminated. Self-reactive and useless T cells. Okay? Central tolerance, as a result of central tolerance in T cell, we have T cells that are able to recognize self-MHC. We have T cells, they are the results of central tolerance, outcomes of central tolerance in T cells. We have T cells that are able to recognize self-MHC. Okay, the T cells that cannot recognize self-MHC, they are eliminated by failure of positive selection. And we have CD4 or CD8 positive T cells that can discriminate between MHC and class 1 and class 2. The CD4 co-receptor and CD8 enables T cell to discriminate between MHC class 1 and class 2. CD4 binds to class 2 and CD8 binds to class 1. And of course, self-reactive T cells are eliminated, but as much as possible, not all of them. It's important. That's why we need peripheral tolerance. So, 10 minutes break and get back to the class in 10 minutes.